Good morning, everyone. I want to start with thank the organizers and the program committee to give me this opportunity to share with you the massSight, the tools to integrate, homogenize, and combine metabolomics data. My name is Ali Rahnavard. I'm assistant professor in the George Washington University. Our research group is part of the Computational Biology Institute under the Department of Biostatistics and Bioinformatics. Okay, so I just want to share with you a little bit about the omics advancements in biology. So there are many omics that we heard about, the single cell, metabolomics, and metagenomics. I used to work more with the metagenomics. And as we see here, over the years, the increase of using these different technologies in different domains of omics. One of them in the top right is metabolomics, and what we see there is, we see the metabolomics recently has been used a lot and rapidly advancing and evolving. One of the technology used is LCMS, and you see LCMS also is increasing and is being used to mostly for biomarker discovery. So we look at the different diseases and human health conditions, and we look at those, how different omics, metabolomics technology has been used in research in different human conditions. So what you are seeing here, the x-axis is the years, and the y-axis is the number of publications, and the color reflects that different technology has been used in the metabolomics in the bottom for different health conditions or diseases. For example, if you look at the COVID-19, we see has started in 2020 for the metabolomics profiling, and you see LCMS has been used. The last bar is small because that's being produced, I think, around like a few months ago. So, and that's going to be hopefully, a large bar at the end of the year. So this figure just shows us that in different health conditions, there are many metabolomics profiles being produced for the same health conditions. And by the way, this figure has been produced with using one of our software, PubSight, that you can use this, define these terms, you can use and reproduce this figure for different research. So the motivation here for our massSight from this figure is that we see there are many metabolite profile has been produced for different health conditions, and we want to take this opportunity that we have many metabolite profiles for different health conditions, and if we can combine them and use them to have a higher power in terms of a statistical power to investigate the biomarkers for different health conditions. So the main goal here is I have worked with the different omics. For example, in the microbiome world, you can combine the microbiome profiles by just concatenating the columns. But in the metabolomics, we know that is not true. So because there are many properties of the profiles that we need to consider when we combine them, especially when we don't know the name of the metabolites, right? And when we have used the untargeted approach. There are many benefits of this software. The main motivation for us here is to combine the profiles and have higher statistical power. But you can use it for comparing the annotations of different software, for example. So when the two software they has been used to process the raw data, and you want to see if they have annotated those metabolites correctly or they are consistent or not. So the massSight combines those. So I'll show how this works, and that these bullet points, they're going to be more clear later if they are not already. So let's just think about the massSight, the input. So I'm trained as a software engineer originally, then I moved to biology later. So the input and output are important for us. The input are two processed metabolite profiles, which we don't have the name for those signals. These are two tables. And the goal here in this presentation is if we can combine those two data sets, and that's the goal. So massSight is a software that does different activities. Today, I'm focusing on the combine, how you can combine two data sets. What are the other activities? So the main one is the auto-combine, which you give it two processed metabolomics profiles, metabolite profiles, and combines them. So the other function is consolidate. When you have metabolites that they have one original, actual molecule in the sample, and they are being break down, or the adducts of that in the end results when we get the profiles. So that's the consolidate. I'm not going to talk about that, but I just want to share that the massSight is like a package that has all this function in it. And the auto scale also is a challenge we have with the metabolomics data, when we process them, there is a lot of batch effects and other properties that they are add to the process data, and the scale is going to using the experimental values, like if you have a QC samples in your data, or you have an internal standards, uses those to scale your data and make sure that the biology to noise is higher. All right, so this is a general format for the omics profiles, like the metabolized data. So we know all the omics data. We have information about the samples that we have processed the data from them. We have some information about the omics that we have measured. The blue part, the dark blue here, they are the information about the metabolomics. So for the metabolomic data, the two main descriptors of the signals are the MZ and RT, mass to charge and retention time. So we can use these two properties plus the intensity to combine two different experiments. So when we say experiments, there are two different studies. So when you have two different studies and you want to combine this data, and you don't have all the annotation to just look at the names and combine them. That's the goal. So how we do that? So we started to process data. So remember, when we talk about the process data, there are many challenges that we assume has been tackled ahead. For example, if they are this metabolites, they have been annotated. So we know that's a big challenge. So that we are not tackling that here. We assume those exist. So we have the data set 1, data set 2. We start with the two filtering steps. The first filtering step is we're going to remove the metabolites that they have an overlap in terms of retention time or MZ. And that way we can reduce the ambiguity when we try to combine the data sets. And you'll see why this is important later. Then after that, we try to see the potential matches between the two data sets. And we remove those that they don't pass some specific threshold that are defined or by user, or automatically the software finds them. After that, the goal of these two filtering steps is to find the drifts in three different descriptors of the metabolites. Number one is if there is a drift in RT between the two data sets, if there is a drift in the MZ, or if there is a big drift in the intensity. So when you have those, so we model the drifts. So this step 3 is to find how these signals they have changed by finding the main trends in the data when we look at those. And if you see, there is in the y-axis we look at the delta MZ, delta RT, and the differences between the intensities. After that, what we do, we model the drifts, and based on those models, we adjust the data set 2, which is the query data set. So basically, we get the descriptors of the second data set very close based on the drift that we find to the first data set. Now we have two data sets based on these approaches for drift modeling. They get very close in terms of how they have been processed. And now we can look at the closest signals among those two data sets, the original data set for data set 1 and adjusted data set 2, that we believe now, based on the modeling, gets very close to what it should be if we look at the data set 1 as the reference. So then we have a process that goes through the data set 1 and adjust the data set 2 to find the closest signals in the two data sets and report what is the main close metabolites to the original data set or the query data set 1. So basically these steps, there are 3 steps. First was we filter the features, we model the dynamic of drifts for those descriptors, and then we score the matches to find the best match for each metabolite in the data set 2 compared to the data set 1. So why this is important? We have many studies in our group that when we look at the drifts and the MZ, RT, and all those descriptors, there is a lot of challenges when you have a lot of samples in one study. So when you have a lot of samples in one study, to scale the study and use the existing software to annotate them or find the signals, that's the one challenging thing. So if you have multiple studies and you want to process them together, that even going to increase the challenges. But here, our approach makes these big studies or multiple studies, combining them after the process steps easy, because you can process the smaller data set easier than very big data sets because of the inconsistency among those descriptors that I mentioned. So basically, our approach, if you think about the naive approach, is if you have multiple studies, the batch files, batch 1 to n, and you want to use one aligner to get all those data together. That's going to be very challenging, especially when the data sets come from different experiments. But our approach tries to use the process data, and you can repeat it as much as you want. So you can, that's why it's scalable. So you can combine 10 studies, each 3,000 samples. So it's very scalable and very quick. All right, so that's the whole approach. One of the advantage of this work is that it's very user-friendly, it's easy to use it, and we have applied in multiple studies, and we have made the result and the code available in our GitHub. So it's an R package, so you can bring your process data. The only thing you need is those three descriptors, like MZ, RT, and the average intensity across all your samples in the study 1, and those information for the study 2, and then you can run it through our package, massSight, and tries to match the data sets and gives you one data set combining the two profiles. So let's look at a few applications here for this study. So I was part of the Integrative Human Microbiome project. In that study, we look at the microbes and in relation to inflammatory bowel disease. One of the omics that we processed in the longitudinal setup was metabolites, right? And there is another study before us, PRISM. Both they were on the same disease, IBD. So we look at that study. So how they look like, you have a different health conditions, non-IBD, UC, colitis, and Crohn's disease. So those are two different types of IBD, and we have it in one study, different centers, they process the data, and we have that study. There's another study before us, 2018, that they did the process, very similar study. If you look at the numbers and the disease types, they are almost exact. And they had a different omics, by the way. They had these two studies. They had the metagenomics data and the metabolomics. Here we are looking at the metabolomics part. So this is emphasized that there are many studies. This is just one example that there are many studies in metabolomics that they do very similar process and with the similar health conditions that they can be combined and use this approach to increase the statistical power. So that's one application here. And that can be used. I'll show the result for that. But there is there are other places that we apply our techniques. I just want to think about the other domains that you can apply these techniques. Here there is the IHMP dataset. It's a very large study. So we processed the data. The original dataset was processed with the Progenesis QI. Is a software to process the raw data and get the metabolites. And then we use the compound discovery, the other software, to see if these two techniques, they are consistent in terms of annotating the metabolites when they give us the profile. So there are many applications for those. Let's look at the first one where we have the IHMP and the PRISM studies, both IBD with the similar conditions and similar approaches as well. So they have used a untargeted metabolites with the 4 different pipelines and they have all the metabolites. So here I put the information that we needed, and these are some visualization. By the way, massSight produces a lot of ggplots too, and they save them as a ggplot objects and PDFs that you can look at those as well. Here we look at, if you look at the PRISM, in one of the approaches used in PRISM, we had 2,316 signals, and when we matched them to the IHMP, this is very important here, we had about 87,000 metabolites, and 2,044 they were matched here. All right, so the accuracy to see how the method works, we look at the annotated ones. So if a metabolite, if the data set 1 has the annotated metabolites and the data set 2 has also the annotated one, which usually are a small proportion, we look if they are matched correctly, and 98% were matched. Okay, so the massSight is available as a package, our package in GitHub. We have a documentation how you run it, and we are actively, helping the collaborators and the users. So if you have any question when you use it, please let us know. So just to summarize, we see the metabolomics data are rapidly increasing, the use of them, and it's good to combine different studies. massSight help us to do that, and has a software and approach. So the software is important for us because it has many modules that help us with the steps, and we get the users' feedbacks and we incorporate them in the software. So it's very easy to use it, and I hope it is if you use it as well. And then we have applied on several studies. I show two of them here, and it works very accurately, and we get those, even when we use the very hard situation, which because of the time, I'm not going to go through them, it still works very well. So, and the evaluation application, those are two main things that we are still working on them. This work is not published as a paper, but is a published software, and it's open source. You can use it. The things that we're going to improve for the massSight, the two main things, we're going to improve our approach. We still see challenges ourselves when we applied on many big data sets. There are challenges that, again, because of time, I'm going to skip them. And the scaling and consolidation, those two also going to be two modules as part of the massSight. So this is my group, and these four research staff in the group are actively working on this. And one of my collaborators, Keith Randall, who is my colleague in the same department, is testing it. So we try to always have one dedicated group collaborator to test our approaches. There are many other collaborators that they are using it for their research. Okay, by that I'm going to end, and if you have a question, I'll be happy to take it.
Thank you for a nice presentation. I can see quite clearly a real use for trying to annotate more, for example, distinct studies, LCMS studies, by combining, in particular with the accuracy you show. You can maybe try to do something about that. But I have more difficulties to see the part regarding meta-analysis based on reanalyzing by combining intensities. Because, for example, you explained what you do for handling batch effects and trying to have comparable intensities. But when you have different studies, there are a lot of a kind of variability that has to be considered, even when you are talking about the same subject, same kind of diseases, etc. So how can it be used after batch effect correction, knowing that it may correct over bias at the same time?
Exactly. So great question. You have a software for that too. So what this does, this software is very specific, as you mentioned. It combines the data sets, right? One column adds to the study for the samples, and it says which study it comes from. The study 1, study 2. When you do that, that's a differential expression step. Let's say we have a software named Tweetiverse. By the way, all these are part of our umbrella Omics, which we have a software for that. That's going to be a statistical approach, that how you define your study. And what I recommend for that, for what we do is, when they are combined and you try to do the differential expression, you want to see if a metabolite has changed in IBD versus non-IBD, right? So you add the study. So now they are combined. Many of them, they don't have a name. You don't know what they are. You assume you have a higher power because you have more samples in your study, the combined study. Then you add the study name, study 1, study 2, because you have those information for your samples that after the combination. You call them like the random effect in your model, so you can do the random effect. We have a software for that is that process with a lot of other things that I'm going to skip again. Is the software Tweetiverse. The name is Tweetiverse, which does the differential expression, and it's all this information that can be piped to that software.
Excellent. Thank you.
All right. Thank you, Ali.
Thank you so much.
Next, we have Oliver Crook, who will talk about hydrogen deuterium exchange.
Hi, I'm Ollie. I'm going to talk to you about inferring residue-level hydrogen-deuterium exchange with a computational approach that I call REX. So first of all, what on earth is hydrogen-deuterium exchange by mass spectrometry? So it's a method to measure the conformational heterogeneity of proteins or protein complexes, so basically their shape and how they move. The way it works is we take purified protein or protein complexes and incubate them in heavy water for a certain amount of time. And during that time, the deuterium in the heavy water will exchange with the hydrogens in the backbone at a context-specific rate, and then we can digest our proteins using a non-specific protease online, and then just go into the mass spectrometer. Now, deuterium is heavier than hydrogen, so we just observe this exchange as a mass shift in our spectra that you can see in the bottom right, and therefore we can calculate some for each peptide a percentage incorporation of deuterium. Typically, this experiment is done in multiple protein states, so an antibody antigen pair, or with a small molecule, or mutants, or whatever you like, and then you can measure the difference between these two states in the way that you have this HDX to say, okay, there's a difference in the conformational state or a difference in the structure. So why do people use this technique? Well, it's kind of complementary to things like crystallography or cryo-EM because it uncovers kind of subtle dynamic and structurally resolved protein behavior. People use it to map epitopes and paratopes, so that's the binding interfaces of antibody-antigen complexes. You can see stabilization, so potentially allostery in small molecule applications. And it's now a fairly straightforward experiment to do. You can actually just set it up with a robot to do most of the experiment. So the approach takes the bottom-up method to mass spectrometry, which is the protein is digested into small peptide fragments. In our case, which is different to normal like shotgun proteomics using a non-specific protease, because we want to preserve that kind of structural information in our protein. But this actually leads to some like complications for this particular kind of data. So we have this problem of redundancy. We have two overlapping peptides that might give us very similar information, but how do we combine maybe some statistical information about these two things, or how do we reconcile, if something is 90% incorporated deuterium and something 70%, like, is it somewhere in between, or how exactly do we deal with that? Peptides can differ in length, so there might be 5 residues or 15 residues. Of course, a longer peptide is going to incorporate more deuterium, but it's also going to cover more structural regions. And so effects are going to look larger, but also more variable. So we need to account for this. If you're looking at differences, one peptide could, say, be deprotected in one example, so a decrease in deuterium, and an overlapping peptide have an increase in deuterium compared to some ground reference state. And somehow this is a contradiction. So one question is, how do we resolve this contradiction, or what does it mean? And in general, our digestion of our protein is going to look very complicated. So something like this, but maybe even for 10, 15 overlapping peptides. So we're not going to try and resolve these things by hand. So what is the kind of apparent solution to this problem is to work at the level of residues. So this is the kind of opposite of what you would normally do in your kind of proteomics, is you usually take your peptides and build your proteins. We want to go the opposite way and take our peptides and build our residues, because it's inherently a residue level process. And that has the advantage of the interpretation is the same for every residue. It's like incorporation for a particular residue is between 0 and 1 or 0 and 100%. And these contradictions and redundancies are just resolved by forcing consistency between some model that you have and the data. So the simplest way of solving this problem is just taking that peptide map, converting it into some coupling matrix, and saying, well, the uptake that I observed for my peptides is just that coupling matrix times these unobserved residue level uptakes. And so typically this is a rectangular matrix, so you can't invert it, but you could do a pseudo inversion, and I guess you're done. But not really. So pseudo-inversion is typically unstable. There's no reason that your deuterium uptake will be 0%, between 0 and 1 or 0 and 100. It could be negative, it could be 130%. It's not clear how we would use kind of replicate data in that framework. It doesn't exploit the fact that it was a time course experiment, like we're inferring rates of exchange, not just any kinds of numbers. And there's no, this is kind of under-parameterized problem in some sense, so we go to typically overfit. And so I'm going to whiz through some 2 decades of people trying to solve this problem, and saying, well, let's propose some model, we look at our data and kind of minimize some kind of deviance between the 2 of them, and it's fairly typical to add constraints, so deuterium uptake must be between 0 and 1. But this might still overfit, and so people introduce some kind of smoothness penalty to say our 2 neighboring residues shouldn't have really drastically different deuterium exchange, and so you can say, well, I'm going to penalize them and encourage them to be small. So that's what that term where it says like lambda, and then this kind of penalty is trying to say. But some kind of methodological issues kind of remain. How much smoothness do we expect for a particular protein? What about uncertainty? And how do we quantify any differences between kind of 2 protein states at the residue level? So Rex is hopefully going to help us solve this problem. And what is this? So it's a Bayesian change point model. That probably doesn't mean much to anyone right at this moment, but the idea is that for each residue, we're going to assume that there's a parametric function in time. So just this logistic equation that we have here, we assume that's true for every residue, each with a kind of residue-specific exchange constant. Now, I've simplified the actual equation for this to something smaller just for illustration purposes, but you can think of every residue has this one parameter you want to learn. And then you can think of this as a kind of spatial problem where I want 2 neighboring residues to be kind of correlated with each other. So we're going to take these exchange constants and assume that there's some unknown function that we want to learn. And this is our latent change point process. So we don't know what it is, and we're just going to have to learn it from the data. So a more kind of graphical description of that is there's some temporal model for each residue, and the parameters of these temporal models are kind of shared between neighboring residues, and we have to learn what those are. But we don't know how many parameters is sensible for our model, and so we try and infer the kind of number of parameters that is kind of optimal, this kind of change points where actually these exchange constants are different, how many, and so on. And we use this algorithm called the reversible jump MCMC. So it's just a Monte Carlo algorithm designed for problems where you have a changing dimension to your model. So the question is, yes, okay, I can infer a residue level process, but who's going to believe you? So we want to design a kind of simple benchmark experiment that could be easily repeated by other people in the field, so it's extensible. We wanted it to be accessible as well, so not require any other kind of specialist equipment like NMR. And we also wanted it to be quantitative rather than qualitative. So what we did is we took just a standard protein cytochrome C and performed our usual experiment. We swapped out the digestion column that we have for 3 different proteases. So we would generate kind of 3 different peptide maps. And what we do is we take one of those peptide maps, so one data from one experiment, train our residue level model or residue level models that we're trying to compare, and then use the other digests as a kind of holdout test sets. So in this way, we're just adding the residue level values back up to make the peptides and then comparing the difference. I don't want to labor on the next few slides for too long, but it's just to say that our method has lower error rates than others that have been proposed by people. But I think the more important thing is it does this despite other methods violating physical constraints, right? Other methods allow kind of non-monotonic like variations in deuterium incorporations. It's not actually physically possible for it to go down. And some of them have like, oh, I'm incorporating negative deuterium at this particular time. So what would a particular application actually look like? So I want to have everything controlled and know what's happening. So this is what an epitope mapping experiment would look like. So I have a protein and I'm binding an antibody, and my question is, which residues is my antibody binding at? So typically on the left-hand side, each point is a residue. In this case, everything's stratified by time vertically, and you can see this kind of general shift towards the left, suggesting that some residues are being protected on the antigen by the antibody. And then the kind of middle panel, where you're basically drawing your eyes towards this kind of like little upside down this kind of sink, where you're seeing these residues that are kind of spatially near each other are again being protected. And it's most clear if you just average over the time dimension of the model and say, well, I think it's fairly obvious which residues in the simulated example my antibody would be binding, and we can scale the pointer, this transparency, to the probability, so we have some sense of confidence in that as well. And obviously we have some level of noise in the experiment. So one application we looked at is these bromodomain containing proteins, so BRD4, they're epigenetic readers, and they're kind of a very common therapeutic target. So BRD4 has two what's called bromodomains. So these are highly conserved helical bundles that you see on the right. IBET151 binds as a non-specific binder of these bromodomains, and it binds so both bromodomain 1 and bromodomain 2. We actually see a very similar kind of, I guess, binding signature for both of them, except that in bromodomain 2, it looks like the BC loop, so that's the loop that sits at the back of this binding pocket, is much more strongly stabilized in this case. And that has to be down to the residue differences in the BC loop, but it's not clear that it's actually binding of the small molecule to one of those residues there, because actually it's quite far away. And the way we rationalize this is actually this is inherently effective, that loop being slightly more flexible in the second bromodomain as in the first bromodomain. So here we get a better understanding of the functional differences between these two bromodomains. They've kind of evolved to have different levels of flexibility despite being highly conserved between each other. Now, I've shown examples where you only have like one or two conditions you want to compare, but I've got my robot, so I can do maybe dozens of experiments. LXR alpha is a liver X receptor. It's an attractive drug target because it's involved in the regulation of the cholesterol homeostasis pathway, and its beneficial effect is attributed to the induction of this cholesterol transporter ABCA1. So when it's upregulated, more cholesterol results in efflux of cholesterol out of the body. But a lot of these drugs that kind of induce this effect also have side effects such as lipogenicity. So the question for drug discovery is, how do we balance the beneficial effects against these therapeutic side effects? So we profile 17 small molecules using this kind of approach, HDX, and this computational pipeline. And yeah, as I said before, pairwise comparisons are kind of typically infeasible. So what we do is we just apply our method, project to residues. So we have a big matrix that's just residues by small molecules, states, and just use principal component analysis on that. And that allows us to kind of quantify the kind of conformational landscape in some way. And actually, the only therapeutically approved molecule kind of sits on its own in the bottom left, saying it's kind of somehow different, and we can annotate these therapeutic properties and see actually which residues are involved in that kind of separate these classes in some way. And the great aspect of this is that the loadings of these PCA really are residues on our proteins. So we can see which residues are actually involved in defining that variability within the data. Just to say that we were a Bayesian approach, and so each of these things is a kind of point estimate in the space, but there's no reason why we couldn't actually look at the variability in those approaches and kind of quantify the uncertainty we have in those changes as well, which is important for drug discovery. And also to say there's no reason we need to do this in an unsupervised way. If we're just interested in separating our molecules' kind of functional properties, we can be completely supervised as well and look for which residues are best at separating our molecules as well. And then we can, like, I guess, target these residues for molecular design further down the line. So just to summarize, REX is a Bayesian non-parametric method for residue-resolved hydrogen deuterium exchange mass spectrometry, has high predictive performance, and we can perform all kinds of single protein differential analysis, large-scale experimental analysis with it, and we learned something about LXR alpha's functional properties and how that links to its conformational states as well. So thank you for listening, and I want to thank these people for putting up with me.
This might be a naive question because I am not very familiar with HDX, but where do you get that initial uptake matrix from?
The initial coupling matrix.
Exactly.
Yeah, exactly. So imagine you have kind of whizzed over. So I'm just putting a 1 in that matrix if my peptide overlaps that residue in my protein sequence.
Okay.
Yeah, so it's just saying that is that residue is contributed to by that peptide. So there's information there. Otherwise, there's no contribution.
All right. If there are no other questions, we will continue with the final talk of this morning session. From Q1, we'll talk about top-down mass spec.
Hello, my name is Joan Zhang. I'm a postdoc in Kodaha Lab in the University of Tübingen in Germany. So far we discussed small molecules like lipids and metabolized peptides, and let's talk a little bit about like big molecule intact protein. And my talk is more about like statistical stuff like force discovery rate. So it's not about like tool implementation, but I think this is a pretty important stuff because now I want to deal with some fundamental. So for the audience who are not familiar with top-down is basically illustrated like this. Let's say this sausage is intact protein, and in bottom-up proteomics, what people just do is like slice it, like cut them into smaller peptides and analyze individual peptides, right? One at a time, and one peptide at a time, and when you go to the protein, we infer protein level information from those individual spectrum. But in top-down, what we do is basically we just analyze the protein as is, and hopefully we can get the more like a bird's eye view information, right? So then why are we doing a top-down over TDP? Because it's cool, yeah, for me. And more than that, we can study proteoforms, so proteoforms, and the different forms of protein produced from gene with variety of sequence variation, splice isoforms, and post-translation modification. So for example, in here, let's say we have a gene in the left side, and in the right side, as like steps go by, we have a bunch of different forms of proteins, right? So we have a bunch of proteoforms with different isoforms, different modification, like glycophospho, whatever, right? So if you see the proteoforms in like big scale, as in top-down, we can get more higher resolution information, and we can more information on a function of each proteoform, which cannot be seen from bottom-up study. And what is the challenge in top-down? Let's look at the signal from peptides, which is in bottom-up. In peptides, usually we see one or two different charge states. Like in here, I saw like charge 1 and charge 2. This is a molecule with mass M, and each charge comes with relatively short isotopes, right? But in case of intact protein with larger mass, we have a bunch of charge states. I showed 3 charge states, but it can go up to like hundreds of different charge states. And each charge state comes with quite complex isotopes, right? So this kind of complex signal structure is quite challenging for data interpretation. And worst case is like we have a bunch of different molecules, proteoform molecules, then we have additional peaks in here. And in reality, we don't have any color. In other words, we don't have any charge. So we need to find out what kind of masses are there from these dense peaks. So we want to first go back to the molecular mass that generated the spectrum, and this step is called spectral convolution. So from the very complex convolved spectrum, we want to go back to the list of monotopic masses that generated the spectrum. And this is a few examples from very simple samples. It only contains 6 different proteoforms, and you see very dense peaks, and these red peaks are the peaks from different charges of the same mass. So we have charge from 27 to 68, and this is another different. And then what is the connection between convolution and identification? And I will show you the typical pipeline for identification of proteoform. And first, what we do is we do the convolution, as I just said, and then we get from raw spectrum, we get the convolved spectrum. And after that, we do the database search, as in the bottom case, and we want to get the proteoform spectrum match, or in other words, PRSM. And some people say this is PRISM, but yeah, that's PRSM. And we also have this unwanted PRSM, false PRSMs, always, right? And what people usually do is people do FDR control at the end of the database search or during the database search by putting like target decoy stuff, yeah? We just remove false PRSMs. And if we forget about the first part here, right? And this is really similar to the conventional bottom-up search. So we know in this case, this FDR control works pretty well. But actually, we do have this step, right? And we should think about this case. Okay, what if we have error in the convolution step, right? Maybe we have falsely decomposed spectrum. And then my question would be, when those falsely decomposed spectrum actually go through exactly the same database search, the upper side FDR control can control these false PRSMs occur from this falsely decomposed spectrum. Right. The false convolution can happen in precursor mass determination and or fragmentation determination. In other words, convolution not only deconvolve precursor, but also deconvolve fragment masses. So this falsely deconvolved spectrum can contain precursor mass error or fragment mass error or both. Right? This is one important point. And one problem with top-down field is that we still do not have any like gold standard data set. We lack this high-quality data set. People try, but still we are not there yet. So when we don't have an important standard data set, how can we evaluate if FDR works or not for this scenario? Okay, so what I did was like this. Let's bring forced deconvolution instead of true deconvolution, and let's make forced deconvolved spectrum on purpose. The first case is the spectrum with forced precursor mass. How can I generate forced precursor mass? Simply by adding like plus minus 10 in random, excluding 0. And this forcedly deconvolved spectrum goes through the database search, generating a bunch of forced PRSMs. And second, in this case, I don't touch the precursor mass, but I touch the fragment mass, and I select large portion of fragment mass, and again add some random masses. And again, we have 4 PRSMs. In the last case, I just shuffle all the masses, precursor and fragment masses. And I wanted to see if they are controlled by the target decoy, if they are controlled. So both PRSMs in each group versus decoy PRSM distribution, they are compared. And if their distributions are highly different, that means we are in trouble, right? So this is the figure for cases. There are many cases, so let's focus a little bit. So this blue guy is the target distribution from just normal database search. So good ones. And this red guy, it's not well seen, but there's red, and this is the decoy from just normal search. And these two cases, say, okay, yellow and purple, they are false PRSMs from the purple is all false masses, that means precursor and false mass, precursor and fragment masses are shuffled, and the green is only fragment. And lastly, the bright blue is the case where false precursor masses are used. We have a bunch of arrows, but we have 2 different groups. So the first group in the left side, actually they are good because they are false and they are supposed to follow this red decoy distribution, and they're actually following it. So it is good. But in the right side, the bright blue case, it is not following this decoy. It is really close to the target. That means they are not filtered by target decoy FTR control. So this guy is the headline. So, and I repeated the same analysis for the bottom-up case. It has quite similar tendency, but their distance between precursor mass error and target were wider than top-down. That means this problem is more serious in top-down. Okay, then now we know this is very bad when we have precursor mass error if their control is not working, but then how often does precursor mass determination error arise? Okay, this kind of precursor mass error, it has been well known in bottom-up society, and people try to correct these errors in chart determination. And in top-down, there's no specific study yet, but people are aware that convolution is not very stable, it is error-prone, and still we don't have any robust error control. That means it should be quite frequent. But we wanted to see direct evidence that actually this is happening quite a lot. Okay, so to see in this case, what I did was like this. So I generated the second pipeline. In this pipeline, I just used different deconvolution method. For the deconvolution method 1, I used my tool, and for the deconvolution 2, I used my friend's tool, basically TopFD, and used exactly the same database search. Okay. And then I have 2 differences, PRSM1, PRSM2, and we take this PRSM1 and PRSM2, and they are controlled, FDR controlled by 1% using target decoy, and I checked the spectra that generated these PRSMs. So from the PRSM1, we have these spectra identified by the pipeline 1, and second, we have identified pipeline 2. And I focused on this overlapping area because if this FTR is working, then that means at least for this overlapping area, they should be consistent by 98%, right? So is this really the case? So this is the flow chart or Sankey plot to show if this is the case or not. So the left side is identified by 2. Yeah, so the overlapping area, we have almost 4,000. And when I check the just protein oxidation, I found that they are consistent by like 96% or so, and they're inconsistent by 4%. Oh, I have number. Okay, good. So that means I have FTR lower bound of 2% because they are 4% inconsistent in protein level, which is good. But as soon as I check their sequence, not just protein fixation, they are highly inconsistent, and their FTR lower bound was almost 9%. So we are in trouble, and that really works even when I check the proteoform. Proteoform meaning like not only sequence but the modifications. So these red guys are inconsistent portion, and then why those red guys arise? So I compared their precursor mass consistency between two different pipelines. So this green in the right side, green are connected to the same precursor mass, and all the red parts are connected to inconsistent precursor mass. So, and when this is for the open modification search, so we have a bunch of inconsistent precursor mass. But when I just did non-modification search, then this number quite reduced a lot, because if we do the non-modification search, incorrect precursor masses are automatically filtered out because they should be matched to the correct sequences. So in this case, we have much higher consistency in any level, protein, sequence, and proteoform level. So in this case, we are in good shape. So it clearly shows that for the precursor error-free identifications, FTR works fine. And also it shows that in the previous figure, we do have a lot of incorrect, inconsistent precursor masses in top-down searches. So now we have checked that this FTR control does not work to control the second kind of false PRSMs, right? And then what should we do? So I would like to suggest that there should be error control in the deconvolution to control precursor error mass, right? And then this error control should be eliminating the falsely decomposed spectrum, and in turn eliminate false PRSMs by that. Or more fundamental message should be maybe we can generate the decoy spectrum that corresponding to this precursor error control, and just bring them till the end and use those guys as the second kind of decoy in the FTR control after the database search. But I think this kind of precursor error control in the deconvolution is more modular and much easier to reduce false PRSMs. Okay, then if let's say we are using this kind of pipeline, then we can control the precursor error masses, and then what is the FTR value at the end? So we know from the previous analysis, we know for precursor error-free PRSMs, FTR via target equal works, right? And with this, we can reduce proteoform level FTR, taking precursor error rate into account. So let's say this is all proteoform IDs, and out of them, we have precursor error-free IDs, right? So in this case, in this gray area, we don't have any true IDs because they have incorrect precursor masses in level of proteoform. And now we have let's say we know the ratio between blue and gray. This is 1 minus correctly decomposed precursor masses over all decomposed precursor masses. Let's say this is 1 minus precursor deconvolution FDR, or denoted simply by 1 minus PFDR. And then, now for this blue guy, we know for this blue guy, FDR via target decoy works, right? So we have FDR TD. In other words, if there is two positive IDs, green guy, the ratio between green and blue is 1 minus FDR TD. It is 1 minus FDR via target decoy. So our goal, proteoform level FDR, is nothing but 1 minus green divided by gray, which can be represented by this illustration, and it is 1 minus PFDR minus 1 minus FDR TD. This is quite simple deduction, But the problem here is that we still do not know what is this PFDR or precursor deconvolution FDR, right? It's not an easy question, and we are working on that. We are working on that, and actually we have our own method, but still there's no publication, so I cannot say this is done. So to summarize, convolutionary precursor mass determination propagate to false positive proteoforms, and they are not well controlled by target decoy method, at least in the proteoform level. And to address the proteoform level FDR bias, we presented a corrected FDR calculation formula, but still we don't have method to calculate deconvolution error, deconvolution FDR estimation, and this is under construction, I would say, and it will be presented in the near future. With that, I would like to thank to my colleagues, my PI, Oliver Kohlbacher, and Ji-Hyun Kim, and all other members in our CompMS team, and our collaborator, Andreas Torre team, Philip, and also in Yale University, Dr. Won-Hyuk Jang. And thank you for the organizers, and I would like to take the question. Thank you.
Thank you, Kiwon. See question here.
Thank you. Very interesting talk. I was wondering, you nicely separated two sources of error. And if I saw your slide correctly, you assumed independence when multiplying those two sources. I'm wondering what your feeling is. Are they really independent, the error in the deconvolution and the identification error?
Okay, so actually, I didn't assume the independence, because if they are independent, I cannot draw this actually. This one thing, but I think in this figure, okay, so they are quite dependent, because if precursor error happens, then every PRSM from that error false, right? So, but if you are asking, what kind of independence are you asking this? Quite unclear.
If you go forward on the slide, or we can also discuss in the break. I thought you multiplied the error rates for the FDR computation further on when you do like the green, gray, blue. I thought I saw you multiplied, and I like on the very last step there. This seemed like an independence assumption to me, because you multiply the error of not happening and not happening.
Okay, I see. Yeah, but it's not independence. It's not from independence. It's from like kind of like exclusiveness. So I started with all proteoform IDs, and I only selected the blue area, which is kind of like error-free for the deconvolution, right? And only for those error-free area, as stated in the above, for precursor error-free PRSMs, FDR via target decoy works, right? So this is not independence. This is quite strong dependency. So I'm not using independency, but rather I'm using dependency, I would say. It's not clear. Yeah, because
We can discuss later.
yeah, because I've been there. I was when I submitted preprint, and I was thinking maybe I'm using unreasonable independence, but I found that no, I'm using dependency. Yeah. So we can talk about it. Thank you.
Okay, I think we have our lunch break now, and I would ask all presenters present in the afternoon to, if they have not done yet, to upload their presentations. Thank you.
We'll be back at 1:20.
Just seeing that you were.
Yes.
steps in the other.
Yeah, but there is actually some people I found.
We have all of this slide now.
Yes, of course.
Yeah.
But I don't
So I think anyway, it will be.
so like.
Is using anyone else from?
What is it? No, I don't.
What's the presentation by Chino?
Uploading, I guess.
So it's not staying here.
It will stay right here.
This is the last speaker.
Perfect. So when speakers come up, I will hop up here and start the presentation because there's some minor settings I have to do. And then I'll go into this speaker mode and work on and I hope you enjoy your lunch. Yes. Leave it in here. So you're welcome to leave it in here, but I will not be in here, so if somebody were to come in, we're not live. Yes.
I think this is.
That's also clover.
Yes.
If you want to check in your language.
This is not serious.
They give you like an app.
It's not happening yet.
Like right next to the.
Yes. That's like a subtle way of reminding you.
Because I wasn't sure.
Why was our lives? Why? Yeah, so you're right, yeah. So my idea was if we had a Slack channel, like previously, so CompMS.slack.com. Where it's basically just the four of us in right now. And 3